Hello, everyone. Today, I'm going to talk about accelerating the production of self-amplifying mRNA vaccines using microfluidics. Well, COVID-19 pandemic, I guess we don't need any introduction. We all have been suffering from the past 15 months or so. So here, the demographic shows that the number of infections across the nations, which was collected on April 15th. Currently, there are four different types of vaccines are under intense investigation. There are viral, genetic, inactivated viral, and protein-based vaccines. Some of them got approved from different regulatory bodies across the nations, and at least 80 to 90 candidates are at different stages of clinical trials, as per a recent WHO report. Particularly, the latest approvals from Moderna and Pfizer BioNTech really demonstrated the potential of the lipid nanoparticle, simply LNP based delivery technologies in nanomedicine. However, the real boost came to the field from the ANPATRO approval, which is a LNP siRNA based therapeutic from Alnylam Pharmaceuticals. Further, the latest data shows that the mRNA LNP COVID vaccines are more safe compared to the traditional adenovirus vector based vaccines. In the later case, the data shows that there is a lot of platelet factor 4 based antibodies, which leads to thrombocytopenia in patients. And that's how a lot of regulatory bodies at that point put a pause on some of the vaccines. Along with these mRNA based vaccines, self amplified RNA vaccines have been also investigated for infectious diseases, bacterial antigens, as well as cancers. Particularly, these siRNA-based vaccines are quite efficacious compared to the traditional mRNA-based vaccines because the self-replication of this RNA in cells. The work at Novartis from Andy Gill and co-workers really triggered the field in this direction. Here, the main objective of this work is developing a safe and efficacious saRNA-based COVID vaccine using our PNI next generation platform, which leads to here we are showcasing the rapid screening of lipids and antigens to derive the lead candidates and demonstrating the particle characteristics in vitro screening as well as immunogenicity in mice models. So here is our nano assembler platform for LNP production. Here we can see that we can use our technology from drug discovery phase to all the way clinical scenarios. We can generate the LNPs in 250 microliters to all the way liter scale, which is ideal for clinical scenarios. So I'll quickly go through the methodology, what we used in this LNP production. We generated saRNA LNPs using our next generation mixers on Ignite and Blaze instruments. We use Ignite for bench scale and Blaze for scale-up protocols. And here I want to highlight that we use our PNI proprietary ionizable lipids for our work. The downstream processing of these formulations was performed either on emicones or tangential flow filtration systems. And finally, they are sterile filtered and stored in cryobuffer at -80. We did quite a bit of in vitro as well as in vivo screening, but due to the time constraints, I'm not going to all the details. I would like to show the key data which was collected from mice models recently. So here we can see that our vaccine candidates showed both humoral as well as T cell responses in mice models. The left-hand side, this figure shows that candidate 1, 2, 3, which were generated on Ignite instrument, showed SARS-CoV specific immunogenicity. We could see a good amount of IgG titers in mice serum. We can see a similar fashion with candidate 1 scale-up, which is shown in yellow circles here, and it was generated on Blaze instrument. And also, you appreciate that there is a hike in response from uh, prime to boost administration. And further, our vaccine candidates didn't exhibit any cytokine storm, which is a good sign to develop a safe vaccine candidate. Coming to the T cell responses, our vaccine candidates showed CD4 Th1 positive and IFN gamma CD positive T cell response in mice models. Here on the right hand side, we can see that positive CD4 T cell population. On the left hand side, we can see Th1 immune response data we demonstrated in IFN gamma and IL4 ratios. So both scale up and lab scale candidates showed similar trend in the case of immunogenicity studies. With this, I would like to conclude my talk. Here we showcased the selection of saRNA LNP vaccine candidates using our PNI nano assembler technology. Our vaccine candidates, which are encoded with full-length spike protein antigens, showed very high SARS-CoV specific IgG titers. We used 1 gram dose for these studies, and also we observed 10 to 100-fold hike in response from prime to boost administration. As I said, our vaccine candidates show a very strong CD4 Th1 and IFN gamma CD positive T cell responses. Finally, our nano assembler technology is so versatile, such as that the critical quality attributes such as size, PDI, and encapsulation efficiency, as well as immunogenicity and T cell response were retained from lab scale batch to scale up batch. So with this, I'll conclude my talk, and I'm happy to take any questions. Thank you for listening.